Hello students. In this video lecture, we are going to study about structure of proteins. This is a part from topic biomolecules. The shape of protein molecules are the result of four levels of structure. We are going to study here about the structure of protein and there are four levels for the structure of proteins. These proteins are a very important part of the biomolecules as these proteins are responsible for variety of functions in organism. Proteins of hair, muscles, skin gives shapes to the structure, and while the enzymes, these are the proteins which catalyze physiological reactions. These different functions of proteins are understood by studying the four level structure of proteins, and these four level of structures are primary, secondary, tertiary and quaternary structures. Means all they are differ by only their structures, or what you can see here, the protein structure is primary, secondary, tertiary and quaternary structure. Proteins are known to break down into peptides in stomach and duodenum under the influence of enzyme pepsin being one of them which is secreted by stomach. And the polypeptides are further broken down to alpha amino acids. And this explains that proteins are formed by connecting alpha amino acids. In the last video also, we are studying how these proteins are connected with the alpha amino acids. And the bond that connects alpha amino acid to each other, that is called as the peptide bonds. So the proteins, they contain peptide bonds. So in the structure of proteins, proteins are polymers of alpha amino acids and they are connected to each other by peptide and dipeptide bond. See, these are the amino acids. Glycine, alanine, these are the amino acids. And for the by the removal of H2O, the removal of H2O, OH from this acid and hydrogen of this NH2 group, they form the removal of H2O, and what they form here, the linkage C double bond O NH. So this is called as the peptide linkage. When these two amino acid, glycine and alanine, by the removal of H2O, formation of a peptide linkage, that means formation of a protein, and it creates a structure for protein. And when carboxylic group of glycine combines with the amino group of alanine, what they form here? Dipeptide. And this dipeptide structure is called as the glycyl alanine. So this is the formation of protein. If third amino acid combines to the dipeptide, the product is called as a tripeptide. Like that, when the more these amino acid combines, more than 3 amino acids, they form the tetrapeptide, pentapeptide and hexapeptide. When the number of such amino acids are more than 10, then that product is called as polypeptide. A polypeptide with more than hundreds amino acids, what they form, that molecular mass which is higher than 10,000 mu, and this is called as protein. Polypeptides with fewer amino acids are likely to be called proteins, and if they ordinarily have a well defined conformation of a protein. See her example, insulin, and which contain 51 amino acid and which is a protein. First, we see about the primary structure of protein. Primary structure of protein is a sequence of constituent of alpha amino acids. Means all these alpha amino acids combine together or they form primary structure. Any change in the sequence of amino acid, they results in a different proteins. Primary structure of protein is represented by writing the three-letter symbol of amino acids and as per their sequence in the concerned protein. The symbols are separated by dash. And according to the invention, the N-terminal amino acid residue is written to the left side and carbon terminal amino acid, they are written to the right side. We will see in the next slide. See here, this is the representation of primary structure of protein. These are the amino acids, alpha amino acids, N-terminal amino acids and carbon terminal. Means now see here that NH2 group which is written to the left side, whereas C double bond OH group, it is referred as a C terminal, means carboxylic group, and it is written to the right side. And all these amino acids which are linked here by the peptide bond, peptide linkage, they are represented by the dash and these three letter symbol for the proteins. These three letter symbols for the protein, they are separated by here dash, and this is NH2 to the left side and C double bond OH group is written to the right side. So this is a representation for the primary structure of protein. So here this is the primary structure of protein. All these amino acids, they are in a chain. Okay, they have one or more polypeptide chain. Each polypeptide is in a protein has amino acid linked with each other in a specific sequence. And in this sequence of amino acid, they are said to be the primary structure of protein. If there is any change in the primary structure, that is the sequence of amino acid is changed, it creates a different protein. So this is about the primary structure of protein. For the secondary structure of protein, it shows the two structures. They are found to exist in two different type of structure. One is the alpha helix and one is the beta pleated sheet structure. These two structures are arise due to the regular folding of the backbone of the polypeptide chain due to the hydrogen bonding between the C double bond O and NH. See here, this is a alpha helix structure. It is the most common way in which the polypeptide chains, they are show here hydrogen bonding. See here C double bond O, and this oxygen is bonded here with the hydrogen of NH amine group. Here also see C double bond O NH group. So the bonding is here takes place, and with the NH group of each amino acid received hydrogen bonded to the C double bond O of an adjacent turn of the helix structure. So this is for the alpha helix structure. See here that primary structure is here. It shows the some coiling or looping of the primary structure. It creates the secondary structure. This is for the beta structure, or we can say here the beta sheet structure, in which all these peptide chain are stretched out to the maximum extension here, and they are laid side by side, which are held together by intermolecular hydrogen bonding. These are the two different sheets here. NH, which is bonded here with the C double bond O. Okay, so this is, it forms the intermolecular hydrogen bond. And here this structure is resembles the plated folds of the drapery, and therefore it known as beta plated sheet structure. So this is the secondary structure of protein and beta structure. For the tertiary structure of protein, it represents overall folding of the polypeptide chain, that is further folding of the secondary structure. If primary structure folds, it forms secondary structure. If there are more folding in the secondary structure, it forms the tertiary structures. Means it gives rise to the more complicated structure, like here the two major molecule shape, namely the fibrous and globular proteins, they form this tertiary structure. The secondary and tertiary structures of protein are hydrogen bond or disulfide linkage, van der Waals, and electrostatic force of attraction. So this is for the tertiary structure of protein, which shows the folding in the secondary structure. This is for the quaternary structure of protein. More complex structure, more looping, or what we can say the more coiling is takes place in the structure of the protein. Some of the proteins are composed of two or more polypeptide chain, and they are referred as the subunits. And the special arrangement of these subunits with respect to the each other is known as quaternary structure. So more looping, more coiling takes place in the quaternary structure. So these are the four structures: primary structure in a sequence, secondary structure, a regular substructure. This is for the tertiary structure, three-dimensional structure, and quaternary structure, complex structure of the protein molecule. We can well understood by see these simple structures. Primary structure in a sequence, all these amino acids are linked together in a sequence, and in the primary structure looping is takes place. Secondary, more looping is takes place, tertiary structure, and a very complex giant structure for the quaternary. So these are the four levels for the protein structure. Next concept, we are going to study about the denaturation of protein. Proteins are found in biological system with a unique three-dimensional structure and biological activity. This is called as a native protein. And when these native that proteins are in a native form and they are subjected to the physical change, like change in the temperature or in the chemical change like change in their pH, the hydrogen bonds are disturbed, and due to this these proteins they get unfold and helix get uncoiled, and proteins lose its biological activity, and this is called as the denaturation of proteins. Means after heating or by changing their pH value, what happened, protein lose its property, and protein lose its all this biological activity, and this is called as the denaturation of protein. During this denaturation of protein, secondary and tertiary structures are destroyed, but primary structures remain same. Remember here, by the denaturation process, as there is a disturbance or destroy of the structure takes place, but it is takes place only in the secondary and tertiary structure. Primary structure is not affected by the denaturation process. Coagulation of egg white on boiling or curding of milk, which is caused due to the formation of lactic acid by the bacteria present in the milk. These are the example for the denaturation of protein. So here the diagrammatic representation for the denaturation of protein. This is the protein native conformation for the tertiary structure, and on heating uncoiled and disturbed of this protein structure and it form the denaturation process, and after that uh, on that heating, coagulation or denaturated protein which is formed here, okay, which is a biologically inactive protein. Okay, after the denaturation, what we get, this is the biologically inactive protein, and protein which we are having here in the native form, which is in a biologically active form or which is a functional protein. But after the denaturation, it form inactive protein. So this is all about the structure of protein and denaturation of protein. Thank you.